Hello everyone, my name is Rubén Chazarra-Gil. I'm a PhD student in the Transcriptomics and Functional Genomics Lab at the Barcelona Supercomputing Center in Spain. And I'm going to be presenting my latest work, querying splicing differences across human populations at single-cell resolution. So what is alternative splicing? Alternative splicing is a mechanism of gene expression regulation which contributes to transcriptomic and proteomic complexity. This is a process by which the different combinations of the exons in the pre-mRNA stage leads to different mRNA variants, which in turn can lead to different proteins with different functions, localization, or other sort of properties. Alternative splicing has been widely studied and shown to be influential in biological processes such as development, and for instance it's very illustrative the example of the titin gene, which is the longest protein we find in our body and is responsible for conferring elasticity to our heart cells, and it's very graphic how at the early stages of development the isoforms that are more abundant from this gene are those that are longer and thus confer more elasticity to cells, and as we develop, the higher presence of longer isoforms is balanced with also a higher abundance of shorter and thus less elastic isoforms. Even we achieve the situation if the splicing machinery of this gene fails, the overaccumulation of short isoforms can lead to cardiomyopathies. And another interesting example where alternative splicing is influential is in immunity. Many genes that regulate immune function have been shown to undergo alternative splicing, and also some functions such as T-cell activation or isoform repertoire remodeling during monocyte activation are examples of alternative splicing influence in immunity. In a recent study from our group, we queried gene expression and splicing inter-individual variation across different human tissues and taking into account different demographic traits. And we found, interestingly, that the majority of the inter-individual splicing variation within each tissue, except for some exceptions, is due to ancestry. Following on this finding and considering that alternative splicing profiles can be cell type specific, we wondered if we could find such alternative splicing differences at the single cell level. Hence, our goal is to query splicing differences between human populations in a context of immune response and at single cell resolution. In order to query alternative splicing variation between populations at the single cell level, we have to rely in droplet-based protocols, which are a set of technologies that enable profiling the transcriptome of up to hundreds of thousands of individual cells, and more precisely is the 10x Chromium commercial platform, the platform in which we have enough sample size so to study such differences. But if we want to study alternative splicing with this sort of data, we face some challenges which are inherent to the data. The first one is the data sparsity. This is we have a large number of dropouts, as you can see here where we plot the number of genes versus the number of counts for each cell, which is in this plot a dot. For the majority of the cells, we are not able to detect more than 2,000, 2,500 genes, so this is a limitation. And the second challenge we face if we want to study alternative splicing with this sort of data is the read positional bias. These protocols generally sequence either the 3' or the 5' end of the transcript, and as you can see here illustrated for this ribosomal protein which has this number of isoforms, we can only sequence, or we can only have coverage for this 3' end of the transcript, which in this case is a problem if we're trying to identify the different isoforms, because this region is common to all the isoforms. Hence, the first question that we do ourselves in this study is, can we quantify isoforms with 3' single-cell RNA-seq data? And for this, we propose a simulation-based benchmark in which for each isoform in each cell, we simulate reads considering this 3' bias, read bias that we were mentioning. Since we know the number of reads per cell that we are generating, this is going to be our ground truth. And then we quantify the simulated reads with two popular single-cell gene expression quantification methods, which can be tweaked to quantify isoforms, and this is going to be our test set, the quantification with these two methods. Now we compare the ground truth to the test, and we compute a series of metrics to determine the power of methods to quantify isoforms. One of the first conclusions that we get from our benchmark is that isoform quantification in 3' single-cell data is pretty challenging. Here we are plotting the precision, which reflects the number of false positives in the quantification of isoforms, and we can see one of our method does not detect false positives while the other one does. And if we focus on the recall, which reflects the number of false negatives, we can see that in both cases there are a high number of isoforms that are being present in the ground truth, which are not detected by our methods. And if we perform pseudobulking, this is the aggregation of counts per sample and per cell type, we see how the number of false negatives is reduced. Next, we zoom in into the expression profiles, and by binning the features by their expression on the log2 scale, we can see that the limited recall that we were observing in the overall picture mainly comes from the lowly expressed features. And also, if we focus on the highly expressed features for both methods, we have almost perfect precision. This would mean that all the isoforms that we are calling as expressed are indeed expressed in the ground truth. So overall, some of the insights from the isoform quantification benchmark that we have performed is that in general it is a pretty challenging task when we use 3' single-cell RNA-seq data. That recall is low mainly for lowly expressed isoforms and increases with the expression of features. We also see that precision for the highly, if we exclude the low boundary of expressed isoforms, is close to perfect, and in general we see that aggregating counts improves the detection of isoforms. Now our next question is, can we find splicing differences between human populations? To answer this question, we are going to use a dataset by Haley Randolph et al. From 2021, which is composed of 90 individuals of mixed European to African ancestry, from which blood was extracted and the peripheral blood molecular cell fraction was extracted for culture, and it was cultured under two conditions, one with non-infection and the other one with influenza A infection. This leads to a total number of 230,000 cells, which we classify, or for which we consider the 5 major cell types, and given the results of our benchmark, we decide to pseudobulk the profiles per donor per cell type in order to have more power in isoform detection. We will be performing differential transcript usage, which is a type of analysis that compares the relative abundances of the expressed transcripts between conditions. This is, how do the relative fractions of these transcripts differ between the question of interest. And in order to do so, we're going to use MANTA method, which performs multivariate tests of such fractions between the conditions of interest. The results I'm now going to show are preliminary and are still under expansion. And as an initial proof of concept, we were interested in querying the differential transcript usage differences upon infection. This is because we know from previous studies that one of the most responsive cell types to influenza A infection should be monocytes, and this is the signal we were expecting to get. And indeed, when we model relative abundances as a function of infection, what we observe is that the leading cell type in number of DTU genes is CD4 T cells, and this can be explained by the higher number of cells of this cell type, which also causes that the number of detected isoforms and hence the number of genes tested in this test is larger, and this is the reason why we are seeing as a higher number of DTU genes. But immediately after, the other leading cell types are monocytes, which is expected, as we were saying, attending the previous literature. If we focus on the cell type specificity of the genes that vary their transcript abundances upon infection, we observe that this is a pretty highly cell type specific phenomena, although there is some genes that are shared across the different cell types. And two interesting examples we find with differential transcript usage. This is with changes of the relative fractions of their isoforms upon infections, are these two members of the CXC chemokine family. In this plot, we're showing the relative abundance of each of the transcripts that we can find expressed in our dataset between infection status, either non-infected or flu-infected, and you can see how the relative fractions change across the different transcripts. These are two examples of two genes belonging to the CXC chemokine family. In the first case, CXCL is an antimicrobial gene, and the second case, CXCL8, is a major mediator of the inflammatory response. Next, we query our initial question of interest, which is, do we see changes in the relative fraction of isoforms across populations? And what we observe is the signal that we are able to detect between populations is much lower as the infection case, as expected. And we see that in this case, the number of DTU genes found is pretty correlated with the number of cells of each cell type, which, as I was mentioning previously, conditions the number of isoforms that we can detect, and hence the number of genes that we can test in this relative fraction test. Here, the cell type leading is CD4 T cells, followed by CD8 B and monocytes with the same number of cases. Two interesting examples of genes undergoing differential transcript usage between populations are ribosomal protein S23, for instance, and you can see how the relative fractions of their expressed isoforms change between ancestries. And also PWP2 is an interesting example since it enables RNA binding activity and is involved in ribosomal small subunit assembly. Finding genes belonging or related to ribosome function is reassuring because ribosomal proteins have been previously shown to differ in differential gene expression analysis between populations. So in this sense, it's reassuring to find these cases also differentially transcript used. And finally, our third question, which is, can we find populations response differences upon infection? This is transcripts that behave differently upon infection depending on the ancestry. And as we can see here, the signal is lower and also very similar to the population differences. So as preliminary conclusions of our study, we can say that isoform quantification in 3' single-cell RNA-seq data is a pretty challenging task, and this is mainly due to imprecision in the quantification of lowly expressed isoforms. We can also say that we do trust those isoforms that are being called as expressed for highly expressed isoforms. We also see from our data analysis that influenza A infection elicits a cell type specific change in transcript relative abundances. And from our population comparison, we can observe some very interesting examples of differential transcript usage, which is correlated, the signal is correlated with the number of cells, and also partially enriched in ribosomal and ribosome related genes. So finally, I would like to acknowledge Marta Mele, my PhD supervisor, and also Martin Hemberg and Ye-Won Cho, our collaborators, and my entire team for being so supportive and such a lovely people. Thank you very much.